In this session, we will continue our discussion about microbial characterization techniques. Let's review a little bit what we've learned in the previous session. Remember, there are two commonly used methodologies for bacterial identification. The first method is using genotyping techniques, such as PCR or DNA sequencing, and the second method is using phenotyping techniques, such as biochemical testing. We've also learned several carbohydrate utilization tests, which are usually used to identify carbohydrates fermenting microorganisms and are based on their ability to ferment glucose, sucrose, or lactose. Now, in this session, we will learn the other types of biochemical tests. The first one is citrate utilization using Simon's citrate agar test. And then the second one is amino acid decarboxylation using urea hydrolysis broth test. And also we have sulfur indole motility test. And lastly, catalase activity test. Let's start with the citrate utilization using Simon's citrate agar test. In the absence of fermentable glucose or lactose, some microorganisms are capable of using citrate as a carbon source for their energy. That's why this method is usually used to determine the ability of some microorganisms that can use citrate as its sole carbon source. For instance, in the differentiation of microorganisms from Enterobacteriaceae family. And following the incubation, we can observe the change of color in the media. Blue means positive, while green means negative result. Some microorganisms also can produce ureases, which is an enzyme that is especially helpful in the identification of Proteus vulgaris. Although other organisms may produce ureas, their action on the substrate urea tends to be slower than that seen with Proteus species. Therefore, this test is commonly used to rapidly distinguish the small number of urease-positive enteric bacteria from other non-lactose fermenting bacteria. In this test, positive result is indicated with intensely deep pink color, while yellow or orange color indicates negative result. The sulfur indole motility test is used to determine the ability of microorganism to do several things: reduce sulfur, produce indole, and swim through the agar or be motile. And as you can see on the picture, blackening medium along the step line indicates sulfur positive result, whereas in indole production test, a positive result is indicated by a change of color in the surface after the addition of Kovacs reagent. And for a motility test, a positive result is indicated by a diffuse growth inside the medium. And if the growth is only observed along the step line, we can conclude that the organism is non-motile. Our last test is the catalase activity test. This test is used to determine the ability of microorganisms to degrade hydrogen by producing the enzyme catalase. This test is commonly applied for biochemical differentiation of catalase-positive Staphylococcus and catalase-negative Streptococcus, as well as members of the Enterobacteriaceae family. A positive result is indicated by the presence of bubbles on the tested sample, as you can see here on the picture. Finally, you can learn more about the procedure of each test by watching these videos.